This is the first lecture in the new unit. So this is going to be about central dogma of molecular biology. And this lecture is going to focus on two things: DNA structure and the replication of DNA. You should have already downloaded these notes. If you haven't, you might want to take a second to do it so you can follow along with your own notes on a double screen. So this unit is all about how the genetic information held in your chromosomes translates into physical and physiological traits. It's all about what makes you, you. Before we get started, we're going to watch a short clip about what is a gene. Some of this might be a review for you. I think most of us know what genes are. But as we watch the clip, you are going to answer these questions. What's a gene? How is it different from DNA? What are genes made up of? The length and the sequence, that's the order, of genes determines what? And then the shape and the size of a protein determines what? Questions 4 and 5 go together. And then question 6, how are all living organisms, at least on this planet, alike? The clip is here also. And then if you're outside the Gwinnett system, it's in the YouTube clip.
Stated Clearly presents, what exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckled gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Different creatures have different genes, which is ultimately why their bodies look and function differently. But one of the many reasons scientists believe all life on Earth is related is that the basic DNA code, the language of A's, C's, T's, and G's, is pretty much the same for all living things. Many creatures even share some of the same genes. You might not be too surprised to learn that humans and chimps, which are closely related, share 96% of their genetic code. But what would you think a lowly fruit fly has in common with a beautiful swimsuit model? Surprisingly, about half of its genes. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. So just to sum things up a bit, what exactly is a gene? A gene is a special stretch of DNA, not the entire strand of DNA, just a segment that codes for something. Each gene is like a unique recipe, which usually tells a cell how to make a protein or a group of proteins. Different creatures have different genes, but all genes are written in the same basic DNA language of A's, C's, T's, and G's. I'm John Perry, and that's genes stated clearly.
Okay, so let's get to it. The next unit test is about the three processes of the central dogma. So the first one is DNA replication, and that's what this lecture is going to include. Replication means to copy. So DNA replication is about how DNA makes a copy of itself. The second process in central dogma is DNA transcription. And this is how RNA copies just a portion of the DNA code and carries that message outside of the nucleus. The third process is translation. RNA decodes the message, translates it into specific proteins. Remember, proteins are the foundation for all biological functions. And so by the end of this unit and for your test, you will be able to take a DNA sequence and work it all the way through the process to a protein. This lecture is going to focus on the DNA structure and DNA replication. So this is a little bit of a review. This kind of ties together certain other units that we've done. So this is about replication, the where, why, when, what process. So remember that replication occurs in the nucleus. And from our cell structure and function unit, we know that the nucleus is the control center of the cell. And we know that DNA is like a blueprint for everything that happens in the cell. From our cell cycle unit, we know that before a cell divides, all of the DNA must be copied. Why? If you didn't make a copy of the DNA, then when it's split, the cells would be different and the cells wouldn't have the blueprint to do the things that cells need to do. So you'd have mutated cells at every division and you would have chaos and death. So it's super important then that during replication and cell division, we make a full copy of that DNA. From our very first unit back in August, we know that DNA is a nucleic acid, a macromolecule, big, and it is made up of the monomer, the small, nucleotides. You can break down nucleotides into three pieces, three components, okay? So we have the D in DNA, which stands for deoxyribose. So we have a sugar, okay? We have a phosphate group, and we have a nitrogenous or nitrogen base. And there are actually 4 different kinds of nitrogenous bases. How are those nucleotides put together? Okay, so the phosphate, and if you did the gizmo, you know this, all right, the phosphate right here forms the backbone, the outer portion of the DNA structure. Okay, so here it is flattened, and here it is in the double helix. Okay, so the phosphates form the backbone, and they also bind the nucleotides, the separate nucleotides, together. Okay, the nitrogen bases form the steps or the rungs of the metaphorical ladder, and then take that flat ladder and twist it, and that's what a double helix is. What's going to be really important, and if you did your gizmos, again, you know this also, you should know that each nitrogen base only pairs with one specific other kind of nitrogen base. Okay. So Chargaff's rule, or the base pair rule, shows you that because of this knowledge that we know that certain nitrogen bases only pair with other certain nitrogen bases, then you can figure out how much of each you have in the total DNA molecule through very simple math. So the thing that you need to know what pairs with what. So let's go over this. DNA will have a 1-to-1 ratio of thymines and cytosines and adenine and guanine bases. And this is because certain bases are always going to pair together. The amount of guanine will always equal the amount of cytosine because you always pair them together. And the amount of adenine will always equal the amount of thymine because they always pair together. Now, guanine plus cytosine plus the sum of adenine plus thymine are going to total 100. So if you start with 100 and use simple subtraction, you can figure out what you have. We are going to watch a tutorial, a tutorial that's going to explain Chargaff's rule. But again, you really need to remember this. A is with T, C is with G when you talk about DNA. Okay? All right, so for Chargaff's rule, you are going to add a blank slide, okay? And you are going to follow along with the tutorial. So you're going to need to pause the tutorial, and you might want to write down on a piece of notebook paper and then transfer it onto a slide to follow along. All right, but the take-home point here is going to be that if you know the percent or the amount of one of the nitrogen bases, you can use that information to figure out the percentages of all of the others. And you can check your math, because if you add them up and it doesn't make it to 100, you've done something wrong. If you add them up and it's over 100, you've done something wrong. Okay, so we are going to watch the Chargaff's rule tutorial. Follow along.
So some of you just can't remember how to do the Chargaff's rule question. So you have to know that G equals C and A equals T. So if I tell you that there's a certain amount of something, let's say we have 30% adenine, and I want to find out how much guanine. So what you have to do is, if there's 30% adenine
we'll just use A. That means there must be 30% T. And 30 and 30 equals 60%. That means 60% of the DNA molecule is made up of A and T. So therefore, if 60% of the molecule is A and T, 40% of the molecule must be made up of G and C. Therefore, it must be 20% G because G equals C. So therefore, C must equal 20% as well. Okay, I'll do one more for good measure. Those of you who are still having trouble, make sure that you just work through some problems to help you. Because if you don't actually work through the problem, you're never going to learn how to do it. Okay, so let's say it's T equals, let's say 18%. Okay. So if T equals 18%, because of the Chargaff's rule, A must equal 18%. And 18 and 18 is 36. That means 36% of the DNA molecule is made up of A and T. Okay? So the rest of the DNA molecule must be G and C. Okay? So just do 100 minus 36. Okay? So you will have 64% of the molecule will be G and C. Okay? And now, since 64% of the molecule is G and C, we have to figure out how much of it is G and how much of it is C. And since they're equal, we just split that between them. So it's 32%.
Pretty simple. Okay, so here are the steps a little bit more slowly if you didn't catch it. I feel like kids are really successful for this once they practice it because it is such easy math. So she used, I think, this same example. If a DNA molecule has 30% adenine, what are the percentages for the others? So you know that if you have 30% adenine, you have to have 30% thymine because they always pair together. 30 plus 30 is 60, right? So 60% of the DNA molecule is made up of adenine and thymine. So how much of the DNA molecule do we have left to figure out? 100 minus 60 is 40%. So we know that 40% of the molecule is taken up by both cytosine and guanine. So you have to split it evenly between the two of them, and you get 20% for each. And then you can just do your math. 20 plus 20 plus 30 plus 30 equals 100. You're good to go. All right. So that's basically Chargaff's rule in a nutshell. You will have some examples of this on your test, and we will also do some practices through quizzes and other classroom practices. So here's a practice. So do this. If a strand of DNA is 35% adenine, calculate the other nucleotide percentages. And there's another example. If a strand of DNA is 12.5 cytosine, calculate the other nucleotide percentages. So do that before you move on. This just summarizes where we've been so far. We've talked about the structure of DNA, and how we can figure out how the nucleotides come together. So you're going to need to know the structure of a nucleotide, how the nucleotides bind together, and how the nitrogenous bases match up. Before getting into replication, do this quick review quiz. You might have to insert a text box because this is just an image. When you're ready, let's move on to the second part of the lecture, DNA replication. So even though we've said this at the beginning of the lecture, remember that DNA is replicated, copied during the cell cycle. And we do this because we want to make sure that every divided cell, body cells, are going to have that complete set of identical genetic information so that they look and function exactly the same. And a single strand of DNA can serve as a template for a new strand. And that's what we're going to talk about. It's going to look a little scary, but it's not. It's pretty simple. We already have the basics of knowing what nitrogen bases come together with other nitrogen bases. And that's really the take-home point. So when we are making a copy of DNA, you have the taking apart part of it. Think of it like a zipper. An enzyme, it's called helicase, but you're not going to be responsible for the name of the enzyme. Just know that an enzyme unzips the DNA, all right? And then the bonds between the nitrogen bases are really, really weak. The bonds that hold the backbone together are really, really strong. But the hydrogen bonds are weak so that the unzipping and the coming together and breaking apart can be really easy. So the taking apart is like unzipping, and an enzyme does it. And then making the new strands. Take a look here. They're coming in on each side, okay? And there is a different enzyme, polymerase, that puts it back together, okay? It brings over nitrogen bases, nucleotides, and binds them to the template. So replication is called semi-conservative. And what does that mean? Basically what that means, it's referring to the fact that when you make a copy of DNA, one half of the new DNA that you're making, okay, so that the new double helix, one half of it is going to be the original strand. And then the other half of it is going to be new. And in the end, you are going to end up with two new double helixes that are genetically identical to each other, and they are genetically identical to the parent strand. So here's a summary of what it looks like, okay? So from the original, it's going to split open, new nucleotides are going to come in, it's going to unzip all the way to the end, and then you're going to have two split double helixes. So think about all the DNA that we have, and that at one time, way back in the beginning, before you were born, you were just a single fertilized egg. And from that single cell, you're you. Okay? So replication is fast, super fast, and it's accurate. So errors can be fixed on site. Not to say that cells don't mutate. As you get older, if you're old like me, more and more of my cells are going to mutate because, I've been around for 51 years. And so my single cell happened a really long time ago. So if you looked at my genetic code, I would have many more mutations and errors than any of you would. Okay. But you're nice and young. And for now, it's your replication is much more accurate, say, than mine. So let's do some practice. Okay. So here's a single strand of DNA. You have two different examples. And so what I would like you to do is below type and use the dashes to keep it straight. You're going to just do the complementary strand of DNA. So remember, A is going to match up with T, and G is going to match up with C. The last slide is just telling you where we've been and where we're going. So remember central dogma, replication, transcription, and translation. And today we talked about structure and replication.